All right, good morning. My name is Andrew Knudten. I'm the CEO of Advanced Medicine Partners, AMP as we like to call it. Thanks for surviving the 3 days and showing up here this morning. I know I'm also competing against patient videos. I know that's why we're all here as well too. But just a little background on Advanced Medicine Partners. We're a spin-out of Jaguar Gene Therapy. So we've spent the past, in my personal sort of career, over 30 years on the innovator side of the table. And that means a lot to us because we've been focused on medicines, designing and developing medicines, and looking at CMC through the lens of one of the most important people, the partners and the customers and the patients and the caregivers. We've spun out recently from Jaguar Gene Therapy, where we were instrumental in pulling together all the IND-enabling work, up to and including all of the tox work and moving that program to the clinic, which will hopefully get into patients second half of this year. And that program is on autism spectrum disorder, SHANK3 mutation. So it's very exciting, and that's what drives us and motivates us. And I would say is one of our very unique differentiators. Our team of 50 loves patients and is passionate about patients as you are. So I think we were born out of, I would say, a continued frustration over, I would say, the lack of competency and capability in the space. And we did some significant market research as well as talked with investors and bankers. And this really verified our view that that the field's really starved for a real solution. And so we felt that stepping into this place would be advantageous for our partners and to help move the field forward. There are a few CDMOs out there that we believe are doing things the right way. I absolutely want this presentation today to be more thought-provoking versus just a direct communication. So I'll go over a few things here. I believe that the recent changes, or the step changes, I guess, that we've seen in the space have allowed us to increase the quality of what's going on and to decrease some of the complexity. I'll touch on the paradigm of the three pieces, which I've noted as the title in my slide around quality, time, and cost. But we believe that we no longer need to sacrifice quality. And in fact, I firmly believe that quality is the linchpin to achieving the other two. I already touched on this briefly. Experience matters. Experience matters. It's of the utmost importance and is something that's a differentiator. And you should be going to suppliers that have that experience and can help accelerate your medicines. So we are fortunate right now to be in an amazing time frame. So there's such a transformative time in medicine, and the environment is really ripe right now. If you look at it from a regulatory perspective, and we had some terrific early discussions, different panel discussions, on AAV versus I would say non-virals and other. I am still very bullish on AAV, engineered capsids, and I truly believe it's an and not an or. And so if you think about the mountains of data that sit with regulators right now, what space is it in? It's in the AAV wild types. And if you start to engineer capsids and start to create non-virals as specific vectors, those things tend to be N of one with regulators. And so will we get there? Will these be great tools for us for repeat dosing and for larger payloads? Absolutely. But I believe they'll be one of many tools in our toolbox. I think the real sobering thought for us as an AMP organization was the realization that I believe we truly failed our field for a number of years. We could do so much better. When you think about the mentality of a lot of folks going towards the clinic, it's more of a minimalist approach. Like what are the few things that we can do, or what's the minimum hurdle to get me to this place? And I believe we did a disservice to the entire field, and we saw that ripple effect with investors and limited success of getting into the clinic, and more importantly, getting these medicines commercialized. How many times have we read stories about maybe CMC being the hang up, having to do a little bit of a do loop or start and work again, but spend another year trying to refine something, while we've got really important advocacy groups, they're pressuring the regulators to move something forward. We should not be putting the regulators in that position. And we are at the point now where we have the technologies and the capabilities to not do that. So a quick example from our recent experience with Jaguar, we've executed a submission for an IND where we put together a thoughtful CMC package, and I'll walk through some of the things that we've employed in that journey, and we received 0 questions on the CMC. How great would that be if we start to live in a gene therapy world where the conversation focuses around clinical endpoints, the patient, what's next, right, and not talking about CMC. So what are the common problems that we see facing us from the CMC perspective? We talk and have heard many conversations this week around suboptimal impurity profiles, and we'll define those a little bit more, but I think we're also oversimplifying this in terms of empties and fulls. Insufficient process yields, and I'll touch on this too from a quality perspective. If we're focused on quality, it will point us right in the direction of going after high productivity to achieve the yields and purities that you need that will also positively impact COGM and cost of goods. Immature analytics. This has been the Achilles heel for many programs going in because they're trying to bridge something that might have come out of academia, or you're satisfied with the initial process, but as you get closer to the IND or finish line, questions come up and you have to deal with that. And then certainly timeline delays due to poor success rates. How many times do you go in and you want to run a GMP batch and people weren't anticipating things the right way and something went sideways, and now that batch is in the bin, and now you're back in the queue again to make your product. And then the rework around bridging that's potentially important later. And again, our focus is doing it right from the first time, but also touch on comparability. If you have the right analytics in place, we need to embrace comparability, and I think we can do that. So the title of the talk really, and the focus was around trying to balance these 3 dimensions which we talk about ad nauseum in the space from a program management perspective, but also from a development perspective. But again, you can't just focus on 1 or 2 out of these. You have to have a balance, and I believe that science has allowed us to balance the 3 of these in the gene therapy space. And so I'll touch on a couple components of this. And as you can imagine, compromising on quality, you may get a short-term win in terms of getting your program or eking it into the clinic, but in the long run, you'll be redoing work or doing work that you didn't complete at an early stage in a rush once you have that amazing data. And that's something that we anticipate as a company, and the good suppliers out there will anticipate those sorts of things. Sacrificing the delivery speed certainly impacts our patients, and so we pride ourselves in being very nimble, lowering those sort of levels of bureaucracy in the organization, and really driving decision making. And I don't know how many times that you've also been frustrated with, hey, we ran this experiment, the data's sitting there, we've got to scrub it, we've got to get it through a couple gates internally before we share it with you. No, the data's the data, and we need to share that. We need to drive decision making rapidly. And the cost considerations are huge, and we think about cost in 2 ways: cost to develop and cost of goods manufacturing to manufacture. And I think the 2 are very much linked. But if you really hammer the quality piece of it, which again drives you towards increasing your productivities to allow you downstream to achieve the purities that you absolutely want and need, then the rest of these things fall into place very nicely. I'll flip forward here too and tell you why I have a lot of confidence in this. So if you think about where we were, 5 to 10 years ago, looking at flat stock and hyperstacks and adherent, and look, I love all forms of technology, and you're not going to find me, bashing Baculo versus HEC versus, I think, again, these are all tools in our toolbox. But if you think of where the industry and the space has evolved, we are personally, and I think the good CDMOs out there are approaching 1E17 purified out of 500 and surely 1,000-liter bioreactor. So what does that mean? That allows you to achieve, or I would say the 1E17 purified, meaning after sampling, everything's in the freezer, this is what's going to your patient. So that's kind of the end batch yield. So you've got higher productivities coming out of your crude harvest, and allows you to actually purify away all these contaminants. And now that just becomes a choice, right? What are you going to do to make sure you're delivering the purest and most potent medicine to patients? And that's been our thesis from the beginning, and that's probably our key differentiator as a CDMO. We're singularly focused on providing the purest and most potent and the safest medicine for patients. We truly believe that patients deserve this level of output, and regulators expect it. And it's our hope that we collectively raise the bar in the industry, and those that can't achieve what patients deserve will actually fall out, because that's what's ultimately going to help our industry and help patients and move this field forward. As I stated earlier, I think 90% or greater functional full capsids should be the ante into the game. And so first off, if you don't know what functional full capsids are, I think that's probably an issue, because that's truly what we strive for. We want the most active capsid that's out there, not something that contains extraneous DNA. Also, people get caught up in empties versus fulls. If empties are still a challenge, you should be seeking to partner with someone else. Empties can be removed, okay? The discussion should be around partials and intermediates. And I'll use the example of, if you have a viral capsid that's going to transduce a cell, I'm far less worried about transducing it with nothing than I am with extraneous DNA that has to be chewed up, or far worse, or more potentially detrimental effects to patients. So I think we need to move that conversation beyond empties and into this realm of ultra purities, which patients deserve. So if you're not getting purities in this range, certainly have discussions and challenge your suppliers around this. I would also touch on analytics. You must have multiple orthogonal methods to tell you what's going on. And even on the empties, fulls, partials side, everything from we had a great TMS presentation, but you've got AUC, you've got deep sequencing, you've got so many ways to look at capsids, and the regulators want to see that. They want to understand that you know what you're doing with your medicine, you understand fully what that dose is, and you're delivering them something that's very safe. And again, focusing the discussion on the clinical outcomes, and you're not thinking about any potential safety events. So where does this leave us? I think AMP, and there are a few others out there that we know of that are really best-in-class teams. And I think talking to folks that have the experience, that they've walked in your shoes, and that's something that we pride ourselves in. Our culture, I would say, is pretty unique. Everyone that we have on our team has skin in the game on this, and and we say this with pride. They're paid very well. They're paid like innovator therapeutics companies, where they're not looking to find that next job out there. They care deeply for patients as you do. So that culture is critical, because we don't want a revolving door of project managers or other folks touching your medicine. We want that continuity all the way through the process. Talking about a robust and analytical platform that I mentioned earlier, we did receive feedback on, very strong regulatory feedback on our submission, not only overall, but the fact that potency assays are so critical from a regulator's perspective. You want to get out of the space where you're testing in animals as soon as you possibly can and have a very strong surrogate to determine potency. So that's something that we believe should be well qualified before you even get into the clinic. And so really demand more out of your suppliers. You can balance those dimensions, absolutely. So I'll talk a little bit about where we are today in North Carolina. And so we spun out of our sort of parent company, Jaguar Gene Therapy, which was based out of Chicago. But our manufacturing, testing, process development operations always existed in North Carolina. And so today we have the capabilities for full-scale IND enabling manufacturing and testing work. We have over 50 analytical methods, most of them qualified for GMP testing as of today. That sits in Durham, really close to the facility that we're building. If you look on the right, there's a nice rendering of our 14 TW facility, which will be co-locating all of our staff into that facility at the end of the year, with GMP manufacturing coming online first quarter of next year. And so I'll leave you with some important thoughts. We're not the only ones that are doing this right. There are a few folks out there that are doing this the right way, that we believe, again, that patients deserve this level of I'd say potency, purity, and safety in the medicines that we make for them. I think we all have the ability to move the field forward the right way, and the less failures that we have out there in this space, and the more successes, everybody wins. And so I'm happy to talk with you. We've got some folks on our team that are here today as well. We'd love to talk with you about the things that we're doing differently to move medicines forward. Thank you.